Good day, everyone. I'm Wing from Impact Exhibition Management Company Limited. We are organizing Medlab Asia and Asia Health together with Informal Markets. Medlab Asia and Asia Health is a trade exhibition for medical lab and medical equipment, which include business connectivity and the scientific conference. For this year, we are running on the virtual platform on the 20 to 22nd of October. For more information on our business matching on scientific conference, please visit our website, medlabasia.com. Today's webinar, you will learn the advantage of technology that can be used in the dengue virus detection kit, tuberculosis M avian complex, in short TB detection kit, and the COVID-19 detection kit. And this webinar also includes the contents of the single-threaded tag hybridization, printed array strip, in short is STH-PAS, and the technology that can help the medical technologists. So first, let's explore this new technology, research and IVD development with Mr. Hitoshi Sakurada, Sales Manager of TBA Company Limited, with the topic of strip detection of PCR tests, TBA business for research and IVD.
Hello, I'm Hitoshi Sakurada, Sales Manager of TBA Company Japan. I'm in charge of export materials to overseas countries. I'm communicating by email and online meetings with foreign partners. Today, I'm happy to speak at this webinar of Medlab Asia 2021 to introduce our product and business for potential new collaboration with you. Our technology is strip detection of PCR test. Recently, PCR test is getting more popular due to COVID-19 testing. Our test is strip detection of PCR test. We are working for two kinds of business. One is material supply to researchers in the world, research business. Another is IVD development in cooperation with overseas partner. TBA is a university startup company established in 2013, 8 years ago, in business incubator building of Tohoku University, located in Sendai City, northeast region of Japan. We have patented technology STH-PAS, and we are manufacturing product used for STH-PAS testing. STH means single-stranded tag hybridization, mechanism of the PCR-based strip test. PAS means printed array strip. Our strip name, it can detect the result after PCR. We are doing two kinds of business, research business and IVD business. For research business, we are supplying RUO materials to researchers in the world. Research use material to various researchers in various countries. This is our current main business. The customer researchers are buying our materials for their research. For IVD business, we are cooperating with partner company in Asia to develop IVD together in partners' country. The details I will explain later. Here is overview of STH-PAS test. I explain the procedure step by step and necessary materials. For PCR reaction, you need primer set. For our STH-PAS test, you need tag primer and biotin primer. TBA supplies these primer set. 4 kinds of tag, 1, 2, 3, 4, and tag is short sequence DNA. For target DNA or RNA of your sample, suitable primer sequence is designed by you or TBA, and TBA synthesize tag primer and biotin primer. Using this primer set, PCR reaction begins and the target gene is amplified. After PCR amplification, you get amplicons. That is double-strand DNA. One side has tag, another side has biotin. And then you took PCR tube from thermal cycler and add latex blue beads and buffer. These latex and buffer also TBA supply. This blue latex is aniline coated, so it binds with biotin of amplicon. Finally, you insert PAS strip into the tube. Amplicons are developing on the strip as lateral flow from bottom to top. On the PAS strip, TBA already printed complementary DNA in line from bottom to top 1, 2, 3, 4 for each tag 1, 2, 3, 4. Due to hybridization, tag of amplicon is captured and accumulated on detection line. This gathered amplicon show blue color by latex blue beads. This is basic principle of STH-PAS. We supply tag primer, biotin primer, PAS strip, latex buffer. Any target gene can be detected by design of suitable primer. This is details of material TBA is supplying. Tag primer and biotin primer are freeze-dried oligonucleotide. PAS strip in this package. Latex, blue beads. Dilution buffer, 4 kinds of buffer we supply. The user can try different kinds of buffer to get better signal of PAS strip. We include these materials in box and ship them by FedEx, export to various countries. Our materials should be kept in refrigerator for long-time storage, but we ship them by normal room temperature because we confirmed all materials are stable for transportation period, several weeks, at room temperature. Even tropical room temperature, 2 months is okay. And I explain the relation between the customer and the TBA. According to customer's order, TBA prepares these 4 materials and supply export. And customer researchers design the primer sequence for their target. Sometimes TBA support the primer design by sequence matching verification on computer basis. And customer optimize PCR condition or isothermal amplification condition. If using clinical specimen, the customer extract the genome from sample and then perform STH-PAS test. So far, our customers are using our STH-PAS for various test targets, various infectious disease testing. For example, dengue, tuberculosis TB, cholera, COVID-19, drug resistance genes, and others. Sometimes TBA support and consult with customers, and depending on customer, TBA can do contract R&D also. In that case, we involve more for customers' research work. These are major applications our STH-PAS test for infectious disease detection. Basically, our collaborator and customers developed these tests. we supply materials. But some of them, TBA also more involved in development. Now, we TBA is not just raw material supplier, and we have knowledge and contents of these tests. Look, various specimens. Top, the sputum specimen for respiratory infection detection. We have TB mark kit. TB is tuberculosis. Mark is M-RBM complex. It's a major cause of NTM, non-tuberculosis mycobacteria. Another kit, we have respiratory infection kit. That is multiplex of these various pneumonia bacteria. Mycoplasma, Chlamydophila, Legionella, Streptococcus, Bordetella. And middle, serum and plasma specimen, we have Dengue subtype at-once kit. That is genotyping Dengue 1, 2, 3, 4, multiplex detection of Dengue 4 subtypes. This typing is getting more important recently for better treatment and epidemic study and analysis. And we have mosquito-borne virus 3 kit. This is multiplex of different mosquito viruses: Dengue, Zika, Chikungunya. And bottom, urine and swab specimen, we have STD, sexual transmitted disease kit. That is multiplex of chlamydia, treponema syphilis, Neisseria gonorrhea, rare plasma. And final, stool specimen, there is drug resistance gene detection for carbapenemase gene, beta-lactamase gene of bacteria from stool. For more details of these kits, in next second session, by now Dr. Sakurada, R&D Director, will explain about clinical evaluation result of TB mark kit, Dengue kit, and the collaborators respiratory infection kit, and isothermal amplification instead of PCR, LAMP and RPA for drug resistance gene detection. Based on such infectious disease test knowledge, TBA is working for IVD development also in cooperation with some IVD kit manufacturer in Asia. Most important project is cooperation with Vietnam Medlab company. This project, TBA supply raw materials for IVD, and in Vietnam, partner Medlab factory manufacturer IVD test kit. Like this. The kit includes premix primer, pass strip, latex buffer, and Dengue subtype kit. We did clinical evaluation and now waiting for registration of Ministry of Health. And TB mark kit and STD kit are on the way. After getting registration, we plan production and distribution in Vietnam and other country too. Vietnam and other country soon. This is summary and geography of our research business and IVD business. Major customers, collaborators, cooperation partners. For research business, TBA supply materials to research institute, for example, St. Luke's Medical Center in Manila, Philippines, Chulalongkorn University in Bangkok, Thailand, Cairo University, Egypt, Institut Pasteur Tunis, Tunisia, Cambridge University, United Kingdom, Virginia University, United States, Most of them are using our STH-PAS for their local infectious disease test. They develop new diagnostic tool for their research. We are very proud we contribute to such meaningful research in those countries. And now we look forward to working with new customer in new country. And for IVD business, we are cooperating with Medlab, Vietnam, for recent years. And we just started little work with a Taiwanese company this year. We also look forward to work for the IVD development in cooperation with new partner. Thank you for listening. This is final slide. I presented overview of the product and current business. We look forward to new cooperation with you. Please listen more details, technical information in next session of now Dr. Sakurada, R&D Director. For any inquiry and questions, please contact us. For research business, please inquire for quote and price of materials and how to use STH-PAS for new detection. Please ask us. For IVD business, we welcome new cooperation for new IVD test development. Please let us know your interest and demands. Please feel free to ask us. For more information, this QR code for our website. Please check it. Contact form of our website or this email address, inquiry@tbya.com. You can reach us. If you send inquiry, we will reply to you. This picture is scenery in a city northeast of Japan, and this is our member of production, R&D, and administration. We welcome your interest and inquiry. That is all my session. Stay tuned to next technical session by Dr. Sakurada. Thank you very much.
Thank you, Mr. Hitoshi Sakurada, for this new knowledge on the detection technology. And next, let us begin and meet with Mr. Yutaka Sakurada, Director of R&D of TBA, with the topic of strip detection of PCR tests, TBA tests for Dengue, TB, and COVID-19.
Hello everyone, I'm Yutaka Sakurada, Director and R&D Manager of TBA. Today, I would like to introduce our technology and products. First of all, this is today's topics. I will introduce these topics from now. First, the advantage of our technology STH-PAS. Using this advantage, we have developed a dengue virus detection kit, TB map detection kit, and COVID-19 detection kit. I will introduce them. In particular, the dengue detection kit has already undergone clinical evaluation and is now in the process of submitting to Ministry of Health in Vietnam. In addition, we are collaborating with Chinese company. So, they have developed a product using our technology and is submitting in China FDA. I will introduce their product as well. Finally, I will share the latest information of our technology. In particular, we will introduce a multi-detection method that combines the isothermal amplification and our technology. Advantage of STH-PAS. First advantage, we called it STH, single strand tag hybridization. In the conventional method, the PCR product is in double strand. As a result, a denaturation process is required to make a single strand for detection. Furthermore, when they are hybridized with the detection probe, competition with the complementary strand occurs and the hybrid efficiency decreases. On the other hand, in the STH method, a primer with a single strand tag is used, and after PCR, this part remaining single strand. So, no denaturation step is required. In addition, there are no competing complementary strands, and during chromatographic expansion, it has the chance to detect all PCR products. As a result, extremely high sensitivity is available. The second advantage is that easy multiple detection. In order to detect multiple targets in immunochromatography, it is necessary to generate a specific antibody for each target. On the other hand, our DNA chromatography can detect multiple targets simply by adding a detection probe. The probe can be chemically synthesized, and the sequence can be easily designed with in silico. So, it is easier than creating a specific antibody. Our technology is easy increase multi-test. The third advantage is that the PAS can be used in common. The target specific primers are used by selecting different tags for each target. For example, in panel A, each target is tagged different for detection. Each target is detected by a tag at a different location on the strip. So it can determine which target is positive. Similarly, different target panel B uses different specific primers for each target, but the same tag as panel A can be used. As a result, panel A can detect the A, B, C, D gene, and panel B can detect XYG gene on the same type of strip. Common strip can be used. Next, I will introduce the dengue detection kit. We will develop two types of dengue detection kit. One is MBV3, which is a kit for detecting dengue, chikungunya, and Zika virus. And the other is a kit for distinguish dengue subtypes. In the first version kit, nucleic acid was extracted using a Kaizen spin column method amplified by RT-PCR and then detected by strips, which required a total of about 3.5 hours. By revising the enzyme, we have shortened the RT-PCR time and upgraded it. A kit version 2 can be detected in a total about 2 hours. In addition, we have also developed a new kit that can be more easily extraction using serum direct extraction reagent. It can detect very quickly for a total of 1 hour, but sensitivity reduced a little. This direct serum extraction method uses a special extraction reagent called cell-ease RNA V2. Cell-ease RNA V2 is a reagent developed for the purpose of extracting genes from viruses containing in serum. Simply mix with serum and heat at 75 degrees for 5 minutes. That can be added directly to RT-PCR. Dengue subtype serum direct detection kit adopted this reagent. The clinical performance data of the dengue kit is shown. This research is the result of using first version kit. MBV3 kit is for detecting dengue, chikungunya, and Zika virus with taking an advantage of the STH-PAS technology. It can detect which virus is infected from serum in a single test. We conducted a clinical evaluation of this kit at the Pasteur Institute in Nha Trang, Vietnam. Samples were collected from suspected dengue patients at multiple hospitals, and NS1 tests of dengue were performed at each hospital. At the same time, the samples were transferred to the Pasteur Institute and measured with the real-time PCR method and our MBV3 kit, and the results were compared. This is the result. CDC's Multiplex RT-PCR kit was used for real-time PCR. When the results were compared, there was a 95% correlation. Compared with the NS1 test, the result at each hospital, the correlation was 54.3% for both the MBV3 kit and the real-time PCR kit. the Kanfa Hospital of Tropical Disease compared the MBV3 kit with NS1 test. The results showed a high correlation of 92%. In conclusion, the MBV3 kit was highly correlated with the real-time PCR kit. The NS1 kit was considered to vary depending on the measurement hospitals. The dengue subtype detection kit. It can identify 4 subtypes of dengue virus: type 1, type 2, type 3, type 4. It's similar to MBV3 kit. The compared our dengue subtype kit with real-time PCR method. It uses the samples that were dengue positive samples in the previous clinical evaluation of MBV3 kit. This is the result of comparison of our dengue subtype kit with CDC Multiplex real-time PCR kit. Our dengue subtype kit showed high correlation with the real-time PCR kit. The same results were also obtained in one case of type 1 and type 2 co-infection. Next is TB MAC detection kit. This kit can detect TB and MAC at the same time. The detecting image is like this. If the sample is TB or MAC positive, TB or MAC detection line turns to blue, and internal control line also turns to blue. Our collaboration company evaluated this kit. The comparison result of our TB MAC kit and Roche Tuckman kit and Mike ID. Compared to Cobas Tuckman, the same results were obtained with samples derived from cultured bacteria and clinical specimen. In addition, the fact that the sample with a very low number of bacteria showing CT value 42.3 in the result of Cobas Tuckman was also detected. It is considered that our TB MAC kit is the same sensitivity as Cobas Tuckman. The last kit is the COVID-19 detection kit, which has spread worldwide since last year and has caused serious damage. COVID-19 detection kit detects the SARS-CoV-2 virus RNA, the detection image like this. We developed detection kit using the primer set found by the National Institute of Infectious Disease, Japan, and compared it with real-time PCR method. As a result, almost the same result as the real-time PCR method was obtained. The sensitivity of our method is comparable real-time PCR. to introduce the product of Chinese company that is developed a detection kit using our technology. This kit detects 6 respiratory pathogens: MP, CP, RSV, flu A, B, and COVID-19. It is made up PCR reagent and a detection device. The detection device is very convenient that just set the PCR tube and close the lid. Also, a major feature of this kit is that it has an anti-pollution system. The anti-pollution system is performed in 2 ways. One is the device that is completely sealed to prevent aerosol diffusion of PCR amplification products. The other is the reagent using DUTP and UNG. The detection flow using device is shown. After PCR, set the tube in the device and the detection operation is complete. The operation is very simple and the device is completely sealed to prevent contamination. The design of respiratory 6 pathogens detection. In this way, 6 types of causative pathogen can be detected. The photo actually detected is shown. In this way, 6 pathogens have been able to detect exactly. This kit is currently in the process of submitting in China FDA. Finally, we will update the technical information. Multiple detections are difficult with the isothermal amplification method. Using our technology, multiple detection can be easily performed even with the isothermal amplification method. I will share the 2 articles information. The first article was our publication, published in Journal of Microbiological Methods. Isothermal amplification is used to detect multiple mutations in the rifampicin-resistant gene in tuberculosis at once. When amplified the RRDR region with LAMP, a primer that recognizes a single nucleotide mutation in the RRDR region is tagged. After amplification, the then detection by the STH-PAS method. The isothermal amplification method has never been used to detect multiple mutations at once. This is primer design. The blue nucleotide shown the RRDR region, and LAMP primers are designed outside this region. 2 types of primers, wild type and mutant type, are arranged for 4 mutations. The detection is performed with totally 8 tags. According to the detection design, the mutant blue band appears shift from each mutant compared to the wild type. The 31 mutant strains have been detected and shown to be accurately result. The second article was published by our clients, published in PLOS One, which is an example of simultaneous detection of multiple drug-resistant genes by RPA, recombinant polymerase amplification. It shows multiple detection using our STH-PAS method. That's all. Thank you for your attention.
Thank you, Mr. Yutaka Sakurada and Mr. Hitoshi Sakurada for your great content. It is our pleasure to host this webinar. We hope that we have provided all our attendees with very useful scientific knowledge today. And thank you, TBA Company, our speaker, and our attendee once again. If you need more information on these products, please visit TBA website and contact with the team. Thank you.